This looks delicious. I was starving. That's weird. My throat feels tight.
Are you okay? You look a little pale.
Help! Someone call an ambulance!
What's going on?
He just started choking and now he's yellow.
I'm sorry. He's gone.
Alright, tell me what happened.
I don't know. He was eating his pasta, and then he just started choking. It all happened so fast.
Did he say anything before it happened? Anything about the food?
He said it tasted weird, but I don't think he asked for anything special. Just pasta.
Did the dish contain anything that might have caused an allergic reaction?
Well, I think it had cheese in it, but I don't remember if he asked for it or not.
Something isn't adding up. This isn't just food poisoning. It might be something even worse.
I just finished up the autopsy, but I couldn't really find anything in his system that would coincide with the poisoning.
Okay, then what could have killed him?
I don't have the slightest idea. Oh, I think I found something. You're looking at a blood clot, a really substantial one.
Please tell me you've got something, Doc.
Oh, I have something, all right.
Oh?
This is not an ordinary case of poisoning. Take a look at this.
And what is this?
Those are red blood cells. They should be round, but these are shaped like crescents or sickles.
And this is important because...
Well, it means that this individual had a genetic condition, and whatever their condition is, it caused their red blood cells to become stiff and deformed.
You don't know what it is?
No, but what I do know is that the condition of their red blood cells made it very difficult for them to carry oxygen. Over time, these cells seem to have caused blockages in the bloodstream. It would have led to pain, organ damage, and in our severe case, death.
So you're saying he didn't die from the food?
No, not directly. But something triggered a severe reaction to his mutation. That's why his skin turned yellow. His red blood cells were breaking down too quickly, releasing too much bilirubin, which is a yellow pigment, into his system.
So what does that mean? What caused it? And what happened because of the reaction?
Well, in our case, a number of things happened very close to each other. Dehydration, infection, stress, and low oxygen levels. My guess? Something in the restaurant triggered a reaction. Maybe he had an undiagnosed case. Maybe the stress of an allergic reaction made it worse.
You said that this was genetic, right? So what exactly is happening here?
Yes. Our case here is caused by a mutation in the hemoglobin beta gene on chromosome 11, which has instructions for making hemoglobin. Hemoglobin is the protein in red blood cells that carries oxygen. A change in the DNA sequence has changed the structure of the hemoglobin, making the cells misshapen. The change in the hemoglobin beta gene causes hemoglobin to stick to each other, and in turn, it makes rigid poles throughout the cell that push against the cell membrane, causing the rigid sickle shape.
You lost me at DNA sequence.
Okay, well, think of DNA as an instruction manual for building a person. You know A bonds with T and C bonds with G. Anyways, when the instructions are copied incorrectly, you get mutations. A genetic mutation is a change in the DNA sequence of an organism. They can be from your surroundings, like smoking or sunlight exposure, or you can get them from your parents. Mutations can be good, sometimes don't do anything, or like this one, be very dangerous for the body.
So the proteins you were talking about before, how do you mess up the structure of them?
Well, to understand that, you need to understand how our body makes proteins. It's a two-step process. This first part is called transcription, where the DNA is copied in mRNA.
Wait, why not just use the DNA directly? Why go through the trouble of making this R, whatever it's called?
Well, since the DNA is like the master blueprint of a building, it's incredibly important that it stays locked inside the nucleus, where it's safe. If it were sent out to the rest of the cell, it could get damaged or broken down. Instead, the cell makes a working copy, a messenger RNA or mRNA, so the original instructions stay intact.
Oh, I get it. It's like keeping the original plans in a vault and handing out copies to the workers instead.
Exactly, and the workers in this case are ribosomes, which use the mRNA instructions to build proteins.
Got it. So how does transcription actually work?
Well, it all starts with an enzyme called RNA polymerase, and it attaches to a specific section of the DNA. Then helicase will break down the bonds between the bases, as it does during DNA replication. But now an mRNA, or messenger RNA, goes to the unwinded DNA and uses the RNA polymerase to build the bases complementary to the bases on the DNA. Cytosine bonds with guanine, and adenine bonds with uracil instead of thymine, since RNA does not have thymine bases. Think of it as puzzle pieces, where the ends have to complement each other to fit.
Uracil?
It's a base. Like DNA, RNA is also made up of bases. Adenine and thymine go together, cytosine and guanine go together. In RNA, thymine is replaced with uracil.
So it's the same message, just with a minor tweak?
Right. Like switching out a letter in a word, but keeping the meaning the same. Once the RNA copy is complete, it detaches and leaves the nucleus, heading toward a ribosome, which is where the proteins are made.
And that's where the actual protein gets built.
Correct. That's the next step, translation. And if transcription is copying down the blueprint, then translation is actually building the structure.
Wait, hold up. You've mentioned mRNA a lot. What even is that? Something similar to RNA?
Well, there are 3 types of RNA: mRNA, tRNA, and rRNA. Messenger RNA, or mRNA, like I said before, is the copy of the complementary DNA that goes to the ribosome. Transport, or tRNA, transports specific amino acids to the ribosome and matches them with their corresponding codons. Finally, ribosomal, or rRNA, reads the mRNA and then uses the tRNA to translate to its corresponding protein.
Oh, so mRNA has the message, tRNA transports, and rRNA puts it all together.
Exactly.
Wait, hold up. What's a codon? And what are amino acids?
So codons are a group with 3 bases that we talked about before. And remember how each base had a base that went with it?
Mm-hmm.
An anticodon is just those complementary bases of the bases on the codon. It's complementary so it can attach to the codon and form the protein.
And all of these things working together are how proteins are made.
Pretty much. Depending on the protein you're trying to make, the codons are different. But all mRNA strands will always start and end with the same codons. mRNA starts with a codon called AUG, the start codon, and then it always ends with a stop codon. Other than that, the codons in the middle can vary.
So, with all the talk about proteins, how is it related to mutations again?
Well, like we just talked about in translation, the mRNA is used to assemble amino acids into a protein. But when there's a mutation, the wrong amino acid gets added. It's like swapping sugar for salt in a cake recipe. Things don't turn out the way they should.
That makes sense. So in our case, a wrong amino acid was added?
Yes, that's right. We have a point mutation substitution. This means that one of the bases was replaced with a different one. It is also classified as a missense mutation, which indicates that the changed base resulted in a different amino acid.
Wait, what's a point mutation?
Well, a point mutation is when a singular base pair is altered, and there are 4 types of point mutations, each being a different way base pairs can be changed. In our case, it's a substitution, meaning one of the base pairs was swapped out for another.
Cases like this remind me that sometimes the biggest mysteries aren't about crime. They're about the human body. One tiny mutation in a strand of DNA was all it took to end a life.
Yeah, and the scary part is, this wasn't something we could have predicted. We don't even fully understand what we're dealing with yet.
Then, maybe we should name it.
Agreed. And we'll need to document every symptom and reaction so we can recognize it in the future.
The name should definitely have sickle in it, since that's what the blood cells look like.
Sickle cell disease. Or maybe sickle cell anemia, since the mutation affects red blood cells and leads to a lack of oxygen.
Sickle cell anemia. I like it.
And now that we know what it is, the next step is making sure people know about it. Early diagnosis could mean the difference between life and death.
So the real mystery isn't just what happened today. It's how many others are out there, living with this, not knowing. I guess some cases don't end with an arrest. Some don't even have a suspect. But every case leaves behind a truth waiting to be let out. And this one? It was written in blood, down to the very last cell.